from the University of Oxford, who'll be talking about tracing Ebola contacts in West Africa.
Thanks very much, Becky. When I was asked to give a talk about contact tracing and other infectious diseases, I began to think about Ebola, tuberculosis, and a variety of other infections, but quickly realized that Ebola would be plenty for a 20-minute talk. And indeed, I'm going to focus on Ebola during the West African outbreak of 2014 and 15. I won't mention the more recent one in DRC because I wasn't involved in that one anyway. The second thing to say, by way of introduction, is that although this is a mathematical modeling conference, I'm going to say very little about the mathematics, rather give some pointers to the mathematicians and modelers, things to think about when constructing mathematical models that include contact tracing. And I also want to say right at the beginning that a lot of what I'm going to talk about is the work of other people. I was privileged to dip into it in West Africa from time to time from my base at the World Health Organization in Geneva at the time. So most of this work was done by other people. Here's an outline then of what I'll say, just a few minutes on each of these things. Contact tracing to trace the origins of the outbreak of Ebola in West Africa. Secondly, to emphasize how testing and tracing, we've already heard this, but testing and tracing is integral to a whole understanding of Ebola epidemiology and control. It's not an optional add-on, it's a central activity. I'm going to say something about what we learned about the epidemiology, something about how effective we can judge contact tracing to be, and then a few remarks about the end game. The picture you see at the bottom of this slide, I took in Freetown in 2015 in Sierra Leone. This was a negotiation with a very forthright and clear-thinking group of women who were essentially saying, we are contacts, but if you want us to stay in isolation behind that tape you see there, then you need to deliver us the following: the food that we need from day to day, the other household items, otherwise we're not going to do it. So Ebola has its reservoir in fruit bats. They're distributed around Central and West Africa, as you see in the map on the left here. And this is the area of Africa that is characteristic of Ebola outbreaks. The origin of this particular outbreak was southeastern Guinea, a village called Meliandu, pictured here. Bottom left, you see the probable tree where the bats harboring Ebola virus were roosting. The bottom right-hand side of the slide is the map of southeastern Guinea, which makes it clear that, unfortunately, in this outbreak, there was a border very close to Liberia and another border very close to Sierra Leone. So although the outbreak happened in Guinea, it was never going to stay in Guinea. It quickly spread to the two other countries, and then it quickly spread, once it had got into those countries, to the capitals of Sierra Leone and Liberia, as well as to the capital of Guinea. So it was right from the start, it was a three-country outbreak. The contact tracing was inevitably complicated from the beginning and involved contact tracing across borders. This is a picture taken from Sierra Leone, looking across the border, looking across the river border, the Moa River, to Guinea. What you see in the foreground is the Sierra Leone delegation welcoming the Guinea delegation, who were wading through the river to have one of the early meetings about how this outbreak was evolving. So right from the start, contact tracing was integral to understanding and controlling this outbreak. Contact tracing was done at the beginning, it was done at the middle, and it was done at the end of the outbreak. It was done throughout. And as other speakers have said this morning, it's immensely time consuming. It requires an enormous amount of effort. But at no time during this very large Ebola outbreak in West Africa, the largest that had ever taken place, at no time did anybody decide that contact tracing was not worth it. The decision was always, it's going to be difficult, but we need to do it if we're going to get this outbreak under control. What you see on this picture here is actually one of the first results of contact tracing, which was tracing back to where the epidemic began. And one of the questions right at the beginning was there only one transfer from the animal reservoir, from the fruit bat reservoir into the human population, or did it jump across several times? And tracing backwards through diagrams like this one, all the black squares have been redacted here. They all included originally names of people. Tracing backwards to the beginning of the outbreak made it pretty clear right at the start that there was only one event, and that was later confirmed by genomic analysis. So this is contact tracing, identifying the origin of an epidemic right at the beginning, where it happened, who was affected, and how the transfer occurred from animals to people. Actually, the precise nature of the transfer, indeed the species of bat, for example, from which the virus came, is still not clear. That's an open question. But contact tracing made it clear right from the start that there was a single origin to this huge outbreak in West Africa. In this slide, I just want to emphasize what others have already said, which is that testing and tracing in various combinations, well, basically it's testing and isolation of the person who's tested, followed by tracing and then quarantining of the contacts. So those four things are really important, and they're integral to the whole of Ebola control. So pictorially on this slide, I've illustrated all of the facets of Ebola control, but whether or not it's case finding of people who are live through diagnosis or dead after the event, whether it's contact tracing that leads to isolation in a specialized facility, or whether it's contact tracing that leads to the identification of a whole community that is then quarantined. Whatever the set of activities, testing and tracing is integral to Ebola control, and it was, as I've said, from start to finish. Now, if you want to do contact tracing, you need people who can organize it. This is a picture taken in Sierra Leone, where the British Army were involved, and they were absolutely crucial. If you want logistics, bring in the army if you possibly can, because they really know how to do it. Of course, the army is often has a difficult presence, in particularly a foreign army, in communities like this. These countries had been previously through civil wars, and the presence of the army wasn't always welcomed in the communities. But the British Army kept a pretty low profile, and they did an extraordinary job of organization, which is central to effective contact tracing. So throughout the epidemic, as I've said, chains like this were drawn. We drew them daily in Geneva based on information that we had from Guinea, Sierra Leone, and Liberia. We did it in the field team did it. So a great deal has been said about using apps in contact tracing. They were used in West Africa in rudimentary form, but not by the means that has been proposed for COVID in the UK and elsewhere. That is, we didn't have mobile phones communicating with each other. Rather, we had a number of outfits that developed various kinds of software which would help monitoring and then presenting the results, constructing the chains. But the level of sophistication in using apps at the moment was nowhere present in the West African Ebola outbreak. They were used essentially as a data gathering and a data presentation tool, as an additional visual and analytical aid, but not with the capabilities that apps are proposed now. And they were never, they never became predominant during the West African Ebola outbreak. They were used occasionally by small groups of people and in small sections. Essentially, the contact tracing was done manually. It was a great deal of footwork. Now, understanding the epidemiology, back in 2015, readers of the economists were instructed as to what the reproduction number was and how you could calculate it, because nobody understood what it was at the time. But of course, everybody in the world now knows what the reproduction number is in the time of COVID. The economist at the time helpfully produced this illustration, taking the basic reproduction number of Ebola to be 2, and then showing how one case led to subsequent cases in generations. The other interesting point about this diagram is that it explains that if you can actually trace contacts and draw these diagrams, you can actually calculate directly what the case reproduction number is. Mostly reproduction numbers are derived indirectly, for example, from the observed growth rate of an epidemic. But if you can do contact tracing, then you can count exactly how many secondary cases come from every primary case. Now, in Ebola, it might be on average around about 2, but of course, as we've just heard for sexually transmitted infections, and it's true for Ebola as well, there's a great deal of variation from one individual to another as to how many secondary cases they generate. And we used various ways of computing this during the Ebola outbreak in West Africa. So one illustration of this is given here. These are data that we gathered together from Guinea. And once we gather together all of these data, actually from, this is from the capital in Conakry, but it applies very widely, we discovered, as is commonplace in infectious diseases, that about 20% of the primary cases gave rise to 80% of the secondary cases. So the 80-20 rule, which has been mooted for many, many years for many infectious diseases, also applied to Ebola under these circumstances as well. But there's another facet of the distribution of cases per person generated, and that facet is with regard to movement. What you see here is an aerial photograph of a part of Freetown in Sierra Leone, where there was, this is the Aberdeen fishing community, where there was an outbreak, a new outbreak of Ebola in February 2015. Incidentally, it's worth saying that although Ebola was happening continuously across 3 countries, every time a case arose and further cases arose from it, it was regarded as a local outbreak and was treated like that. And all cases and all contacts were tracked. But I'm showing you the aerial photo here because once a community like this had been found with an outbreak, there was an attempt to quarantine the entire community. So clearly, if it has a sea-facing side, as this fishing community had, you can quarantine the land side, but it's very hard to stop people leaving on the seaside. This is a picture of what it looked like on the ground. Top left, for example, you can see the orange plastic netting that became a common sight across these 3 countries. This is where whole communities were being cordoned off, and they were told that they had to stay behind that plastic netting for at least 14 days, sometimes longer than that. And clearly there's huge difficulty in maintaining a whole community like that in quarantine. What happened in this particular outbreak is bottom right, you see a picture of the boat, which contained 5 fishermen, several of whom had had Ebola while out on a fishing trip. One of them was dead by the time that they arrived back in the community. And that death in that boat led to several other infections in the community. Next slide shows you how the outbreak unfolded in Aberdeen, but also the consequences that came from that for more widely in Sierra Leone. So these are epidemic curves. They start top left with the point source there, which is the arrival back of the fishermen in that boat, one of whom was dead. The cases that were generated from that you see in the blue bars. But then what happened after that? We and the team in Sierra Leone discovered that about 1 serial interval later, a new epidemic started, a new outbreak started about 100 kilometers away in a district called Bombali, up country in Sierra Leone. And it turned out through contact tracing that that had been started by someone who had escaped the quarantine and had gone home to Bombali. He generated a second generation of cases. Then there was a third generation and a fourth generation before the chain of transmission was eventually stamped out. But contact tracing was absolutely central to that. We learned lots of other things from contact tracing as well. One thing we saw right at the start of the outbreak was that a lot of cases were arising from funerals because dead bodies were extremely infectious. The practice of washing the dead body before burial was commonplace, and many people would get infected at funerals and the events surrounding individual deaths. But we'd asked ourselves, as the epidemic went on, how common was it that people were getting infections from funerals? And the answer was, in the end, that wasn't the predominant source of infection. And this bar chart explains that. The funeral cases, the only funeral cases, are the red bars. The non-funeral cases in blue predominated in the end. So there were many other sources of infection and transmission in the community, and funerals weren't as dominant as we thought they were in the first place. This data came from contact tracing. Now, let me say something about stopping the outbreak, and also about calculating reproduction numbers. This was a time when I was in Liberia, going up to March in 2015. There was one of the last outbreaks in a section of Monrovia, which had been hit really badly by Ebola. But by the end of the epidemic, there was a sense that things were getting under control, and the tracking of chains of transmission like this one depicted on this slide was becoming more effective as well. So what you have here are generations of cases. One original case left, gave rise to 5 more, which gave rise to 10 more. And then eventually, as this outbreak was being shut down through contact tracing, the isolation of the cases that had been tested positive, and the quarantining of the contacts, 10 went down to 6, and eventually the epidemic subsided. So the reproduction number here was 1 to 5, so 5 initially, 5 to 10. That's a reproduction number of 2 in the second generation, and then 10 to 6, so a reproduction number of 0.6 as the outbreak subsided, as all of the contacts had been found. So there's various ways in which we can look at the effectiveness of contact tracing. We assume, as in the previous slide, that reduction in cases was due to all of the activities on the ground of tracking and tracing and quarantining cases. But there are other indications of how effective contact tracing was too. This is actually in the context of the Ebola vaccination trial that was carried out in Guinea. And this is a slide from the summary paper, which was used to calculate in the end the efficacy of vaccination. What happens in this trial is that a case is found, all the contacts of that case are found, all of those contacts are vaccinated, and then you watch what happens through time. Now, some communities have the vaccine and immediately, and other communities have the vaccine a little bit later on. So the key outcome in terms of the vaccine trial was the gap opening up between the lines that you see on this graph later on. So each of the lines on this graph shows the proportion of contacts who developed Ebola through time. And the interesting result, so far as vaccination is concerned, is up the right-hand end of the slide. It's the difference between the green line in the immediately vaccinated people and the red line in the people who were vaccinated later. And the computation of vaccine efficacy from this slide gave efficacy very high, as was well advertised at the time, well above 75% and probably above 90%. But in the context of contact tracing, these data are also extremely interesting with regard to what happens in the gray area towards the left-hand part of this slide. You see, what happens is when somebody is infected, an infected case is found, all the contacts are found, The vaccine can prevent people from acquiring Ebola who have not yet been infected, but all the people who have been infected are going to go on and develop disease. Now, the vaccine prevents a certain proportion of those, but contact tracing protects all of the rest. It isolates, it doesn't protect them from acquiring disease themselves, but it prevents the onward transmission as a result of those contacts being quarantined. Now, to my knowledge, no one has calculated from data of this kind the effectiveness of contact tracing, but in principle, it would be possible from data of this sort. So just to finish, some last thoughts about contact tracing during the end game for Ebola in West Africa. This is a picture from the notice board in the data center in Monrovia towards the end of the epidemic in Liberia. As a motivational factor, every day somebody put a sticker on the notice board with the number of cases recorded on the day before. And clearly what they were looking for is a long series of zeros and no future cases. You can see at the bottom of this picture here, the zeros became common, but there were still odd cases cropping up. And in this epidemic, like in all other epidemics, and this is what will happen in COVID as well, there was a long right-hand tail to the epidemic. So that this chain of transmission that you see here, which is actually the end of the same chain of transmission that I showed on a previous slide, but carried forward through time. By the dates, which you might not be able to read, but towards the end of February, you can see that most of the arrows there are terminal. They're either people who are recovered, labeled with R, or people who have died, labeled with D. But for several days, there were no further cases. And then suddenly another case cropped up as we went into March. And the question is, where on earth did that come from? Now, this happened repeatedly towards the end of the Ebola epidemic, and there were various reasons why secondary cases or final cases like this cropped up. But some of them were routine, as expected. That is, we had missed intermediaries in the chain of transmission. But there were some other interesting explanations as well, which were uncovered by contact tracing. One of those interesting occurrences was the discovery that Ebola virus had some immunologically privileged sites in the human body, which had been suspected before, but never demonstrated as clearly as it was in the West African epidemic. These are the eyes of Ian Crozier, who worked with us on that epidemic. Ian unfortunately contracted Ebola, went back to the United States. He survived, luckily, Ebola after really serious illness. But during his convalescent period, one of his blue eyes turned green. And the reason it turned green is because he was harboring residual Ebola virus in the humor, the fluid behind his eye. And it took him a long time to recover from that episode completely before his green eye, in other words, turned back to blue. And he would have been potentially infectious in a minor way. The virus, it was discovered, didn't flow forward from the tear duct and out into tears, so it wasn't directly infectious, but he might have been. But there was a more important source of residual infection that we came to discover, again, partly through contact tracing. And that is the presence of virus in the semen, male survivors, or presence of virus in semen of male survivors. And this is the summary of painstaking analysis that we did with colleagues from CDC in the US of 13 potential transmission events, which were possibly events of sexual transmission. In fact, in the end, there were 5 of these 13 events that met the criteria for sexual transmission. But what we had discovered at the end of this epidemic is one reason why cases kept popping up is because of male transmission of Ebola to women via semen, in other words, sexual transmission, in a way and at a level of significance that hadn't been appreciated at the start of the epidemic. And this is my last slide. in fact. And it's just to say that in doing all of this contact tracing, other speakers this morning have emphasized that it's actually a huge amount of work, but it's also a huge privilege as well, because contact tracing means making lots of contacts, and there were many extraordinary stories about the way in which people had lived through this epidemic. This is just one picture that I took in Monrovia towards the end of that chain of transmission. This was a household of people who were contacts of cases who died. They were in quarantine. There were 16 people living in this household originally. 9 of them had died. There were 2 elderly remaining, and the household was basically being run by the teenage girl in the middle here. And I asked her at the time, well, what do you want to do when quarantine is over? And she said, I really want to get back to school as soon as I possibly can. And that left me thinking on this occasion, as I did on many other occasions, how would I have coped living under the circumstances that many of these people had lived under? And I think the answer is not half as well as they managed to do it. So a privilege in meeting really an extraordinary array of people who had to live through that epidemic. So I'll end there. Back to you, Becky.
So here's a question from Julia for both Katie and Chris. Learning from STIs and Ebola, what is the one main thing you want to underline for those running the TTI for COVID-19? So maybe if I start with Chris first.
The TTI has got to be made to work for COVID, as hard as it's going to be, because the situation that the UK is in at the moment, as the lockdown gets lifted, is that increased contact that is inevitably going to happen as lockdown is released has got to be compensated for by a control mechanism. And the only compensating mechanism, apart from people maintaining their social distancing to the extent that they can, the main compensatory mechanism is going to be TTI. So it has to work, it has to be made to work as well as it possibly can. And what we saw for Ebola in West Africa is that it's really tough work, but it's tough work that is really rewarded by interrupting chains of transmission. And I think that's what's going to happen in COVID as well. I think it'll be more difficult for COVID than it was for Ebola, frankly.
Made it very clear in his talk, the phenomenal amount of effort that's required to do this kind of contact tracing. I mean, Chris, do you think, given the number of infections we're still seeing in the UK at the moment, how feasible is it that we've got the manpower to do this at the current level of cases?
Yeah, the question is often framed like that, Becky, but I would put it differently. You know, it's commonly been said there are too many cases, we can't do contact tracing. But another way of looking at it is, this is the capacity that we've got. What is the best and most targeted and most effective way that we can operate a testing and tracing system? I would have argued that it should have been done throughout, even though the number of cases had risen to high levels. There is a best answer to the question of how do we do contact tracing, but to whom do we give priority? And I think there would have been emerging answers to that question. So the notion that has been presented, I think by some politicians in the UK, that because we can't do all of what we want, we shouldn't do any of what we want, is really a false argument. The question is really about prioritizing. And it may well be at the moment. I'll be very curious to see how effective the TTI rollout is going to be. And given the effectiveness and the level of compliance and so forth, how that system will be adapted as time goes on.
It's interesting that you talk about prioritization, and I'm sure that will come up again. You mentioned about the 80-20 rule and potential super spreaders, and I wondered how much evidence there is about that in respect to COVID, and how you would how would we even start to decide how to prioritize if we've got limited resources for contact tracing when there are so many cases at the moment?
As was pointed out with respect to STIs, and I'd like to hear Katie's view on this as well. By contact tracing, you get led to, you quickly discover who the super spreaders are, which is important if those are individuals who are continuously infective, as they are with, say, HIV or some other sexually transmitted infections. But there are other important aspects besides super spreaders. For example, we know who is at greatest risk of severe illness with COVID and death from COVID. Older people, people with underlying health conditions. Then that surely must be the focus. They're the people who should first be protected by all of the mechanisms that are available in COVID control, which includes testing and tracing. So if there is limited capacity, then let's see it focused in the areas where it's likely to be most effective. One other really important aspect of Ebola in West Africa, which was illustrated by the Aberdeen and Bombali event, is to have an integrated database where you can track cases who may move large distances, as was very common in West Africa. People often have their homes and living bases a long, long way from where they work. And when they get ill, they go home and they transmit infection. And unless you have a system of comparing data across an entire country, indeed across 3 countries, as we were doing in West Africa, then you're never going to be able to do that kind of follow-up. And this debate has been current in the UK, I know, about centralized versus decentralized monitoring. But I think one of the downsides of the COVID epidemic in the UK, for example, is that there hasn't been an integrated system of data collection, whether it's by apps or by any other means, frankly.
Thank you. This for Chris. Which were the parts which were easiest for Ebola, and are they the same for TB? And I think this brings in, if we can talk about that in framing it with what Lucy said right at the beginning about what is appropriate with these diseases. Where are they most like COVID, where we can actually learn lessons, and where are they less appropriate?
Well, let me just take the TB one, since I was originally going to try and include TB in the talk, but then it would have been twice as long. I mean, TB has often been mentioned in the UK when contact tracing has been talked about for COVID, but frankly, around the world, contact tracing for TB is not routine at all. And the reason is the reason we were talking about a little bit earlier. The given reason is that there are just far too many cases and there's far too much transmission going on in the community, and you can't track people down. But what I'd also said is that it's never not worth doing it, and it's worth trying to identify the circumstances under which it's going to work most effectively. And the areas where in TB it's been very useful is, let me just give you 2 examples. One is that the low-income world is still awash with tuberculosis. There's 10 million cases a year, and TB will kill far more people this year than COVID will, probably. It'll kill 1 million people. But the circumstances under which it's really worth tracing is when you have, for example, a new outbreak of a multi-drug resistant form of tuberculosis, which is distinct from and trackable and distinct and more dangerous than other forms of tuberculosis. And that was done, for example, when we first saw the emergence of what was called extensively drug-resistant tuberculosis in South Africa, and contact tracing was really important under those circumstances. The other circumstance is in places like the UK, where there still is plenty of tuberculosis, but it tends to occur in isolation, in outbreaks that are trackable. And under those circumstances, it is worth doing contact tracing, because it's really worth stamping out a chain of transmission under those circumstances. Those things are really worth doing, but it is more selective for tuberculosis than it is than it was for Ebola and that it will be for COVID, I think.
Right, thank you.